Good morning, students. This is our fourth lecture from chapter number 6, that is Biomolecules. Now, in this lecture, we will discuss about our next topic, nucleic acids. On the screen, you are looking some information about nucleic acids. See that information. A Swiss botanist, biochemist, Friedrich Mitcher in 1869 discovered and isolated nucleic acid from the pus cells. This scientist is discovered nucleic acid from pus cells. That pus cells we are found in pus of wounds. These pus cells we are found when we are facing any kind of wounds. So in this pus, this scientist found nucleic acid. By 1938, it became evident that nucleic acids are of two types, which are deoxyribose nucleic acid, means DNA, and ribose nucleic acid, means RNA. DNA is found in chloroplast and mitochondria also, except nucleus. DNA is the hereditary material in most of the organisms. The nucleic acids are among the largest of all molecules found in living cells. They contain three types of molecules: a carbon sugar, 5-carbon sugar or pentose sugar, second phosphoric acid or phosphates, and number third, nitrogen containing bases, means nitrogen bases. So these are the contents which forms nucleic acid, means DNA or RNA. Now, in 1924, scientist Fulgen showed that chromosomes contain DNA. He showed that nucleic acid contain two pyrimidines, which are cytosine and thymine, C and T, and two purines, which are adenine and guanine, A and G, called bases. Later, scientist Wilkins and its co-workers show that purines and pyrimidines bases placed regularly at a distance 3.4 angstrom. DNA is composed of sugar molecule, which is pentose sugar of deoxyribose type, then phosphoric acid, means phosphate, and nitrogen bases, means nitrogen containing organic ring compounds. Pyrimidine bases are single ring, means monocyclic nitrogen bases, which are thymine, cytosine and uracil, TCU. Purines are double ring, means dicyclic nitrogen bases, which are adenine and guanine, A and G. Scientist Erwin Chargaffs in 1950 shown that pyrimidine and purines always occur in equal amount in DNA, 1:1 ratio. They also found that base ratio A plus T and G plus C may vary in DNA of different groups of organisms and plants, but A plus T and G plus C ratio remains constant for a particular species. So this is the primary information about nucleic acid or DNA. Now we will discuss about the structure of DNA. So on the screen you are looking our next topic, structure of DNA. So here you are looking on the screen now, purines and pyrimidines. that we have discussed on the above topics, purines and pyrimidines. So purines are dicyclic nitrogen bases and pyrimidines are monocyclic nitrogen containing bases. So here on the screen you are looking nitrogen bases, purines and pyrimidines. Now we will discuss about structure of DNA. On the screen you are looking structure of DNA. See the figure 6.11 which shows structure of DNA. The model you are looking on the screen, that model is actually shown or proposed by scientists Watson and Crick. DNA is polynucleotide chain. Nucleotides are consist of sugar plus phosphate plus nitrogen base. Together form nucleotide, and this nucleotides forms polynucleotide chain that we are calling DNA. Nucleosides consist of nitrogen base and sugar, which constitute in formation of nucleotide. So nucleoside means nitrogen base plus sugar. So there are 4 types of nucleosides found in DNA. In a nucleoside, nitrogen bases attach to first carbon atom of sugar, and when phosphate attach with 5 carbon atom. So which forms sugar molecule plus phosphate plus nitrogen bases together forms nucleotides. Phosphate group of lower nucleotide attached with 5 carbon atom of deoxyribose sugar forms phosphodiester bond with that of third carbon atom of sugar. And so these two ends of polynucleotide chain are called as 3' to 5' ends respectively. A single polynucleotide strand of DNA is not straight but helical in shape, and DNA molecule consists of such two helical polynucleotide chains which are complementary to each other and held together by weak hydrogen bonds. In polynucleotide chains or in DNA, A always pairs with T, means adenine pairs with thymine by 2 hydrogen bonds, and G pairs with C always by 3 hydrogen bonds. So here we have discussed some primary information about structure of DNA, about nucleotides, nucleosides, then component of nucleotide, etc. On the screen you are also looking the structure of DNA. Now concentrate towards the screen. On the screen you are looking figure 6.11 which showing you structure of DNA, in which you are looking there are 2 strands which are spirally arranged with each other and showing you a coiled rope like structure that we are calling DNA. Within DNA you are looking AT, GC, GC, and its bonding by hydrogen bonds. Then again you are looking on the top major groove, means large groove between 2 complete turns, and minor groove also between two complete turns, hydrogen bonds, base pairing, etc. So now look at towards right side where they have given some information about the structure of DNA. That structure of DNA or model of DNA is proposed by Watson and Crick. And now see, according to Watson and Crick, DNA molecule consists of two strands which are twisted around each other and forms a double helical structure. The two strands, that is polynucleotide chains, are supposed to be in opposite directions. So end of one chain having 3' end lies beside the 5' end of other chain. One turn of the double helix of the DNA measures about 34 angstrom, and this turn is containing 10 base pairs. So distance between two base pair is 3.4 angstrom. It consists of paired nucleotides and the distance between two neighboring pair of nucleotides is 3.4 angstrom. The diameter of DNA molecule is about 20 angstrom. There are certain organisms like bacteriophages. Alpha X174 is an bacteriophage, and again some several bacterial viruses which possesses its genetic material in the form of single stranded DNA. So here we have discussed the structure of DNA, about nucleotide, nucleoside, types of nucleosides on the basis of nitrogen bases. So in this lecture you have learned about DNA, nucleotide, nucleosides, nitrogen bases, how DNA is formed, components of DNA, etc. So again, take your textbook, read this topic properly, draw these diagrams of nitrogen bases and structure of DNA, and if you are getting doubts, then ask to your subject teacher. Thank you.